So you want to learn about reception, transduction, and response?
Nah.
Not too bad.
What I mean about reception, transduction, and response is we're going to be learning about cell communication. For this, we're going to be learning about G proteins, secondary messengers, and the activation of an enzyme. But let's take this in parts. First, the reception. So we got this little guy right here. He's called the G protein receptor. And if we take an even closer look at it, we can see signal binding sites inside its membrane. Pretty cool. And as a side note, this is where the signaling molecule binding site is located. And this is a segment that interacts with the G proteins. But what does it do? Well, we need a G protein holding his GTP. Right now, he's inactive, and so is the G protein receptor, until a signaling molecule comes out and wakes up the G protein receptor, which then the G protein comes up and touches the receptor and gets a spicy drink of GTP. Once the G protein has its electric drink, it goes over to the transduction and activates that. While doing that, the G protein will lose its GTP and regain GDP. Next is a transduction. For this, we're going to be talking about the secondary messengers. Secondary messengers are usually small non-protein molecules or ions that can relay signals inside a cell. For example, cyclic AMP or CAMP, calcium ions, Ca2+, I'm not even trying pronouncing this, AP3. Their job is to relay a signal to a cell's interior in response to a signal received by a signal receptor protein, or G protein. Instead of explaining what all the small non-proteins do, I'll only explain one. That would be the CAMP. What is CAMP? It comes from, I'm not even gonna try to pronounce that, C-clase, which converts ATP to CAMP, and then CAMP is able to activate protein kinase A. And once protein kinase activates, we get our cellular response. Or, for this instance, our enzyme activates.